Protein biosynthesis basic details. Protein biosynthesis is the process in which cells build protein. The term is sometimes used to refer only to protein translation, but more often it refers to a multi-step process beginning with amino acid synthesis and transcription, which are then used for translation. Protein biosynthesis, although very similar, differs between prokaryotes and eukaryotes. The events following biosynthesis includes post-translational modification and protein folding. During and after synthesis, polypeptide chains often fold to assume the so-called native, secondary and tertiary structures. This is known as protein folding. Introduction. Protein biosynthesis refers to the process whereby biological cells generate new proteins. It is balanced by the loss of cellular proteins via degradation or export. Translation. The assembly of proteins by ribosomes is an essential part of the biosynthetic pathway, along with generation of messenger RNA, mRNA. Aminoacylation of transfer RNA, that is tRNA, co-translational transport and post-translational modification. Protein biosynthesis is strictly regulated at multiple steps and error checking or proofreading mechanisms are in place. The cistron DNA is transcribed into a variety of RNA intermediates. The last version is used as a template in synthesis of a polypeptide chain. Protein will often be synthesized directly from genes by translating messenger RNA. When a protein must be available on short notice or in large quantities, a protein precursor is produced. A proprotein is an inactive protein containing one or more inhibitory polypeptides that can be activated when the inhibitory sequence is removed by proteolysis during post-translational modification. A preprotein is a form that contains a signal sequence, an N-terminal signal peptide that specifies its insertion into or through membranes, that is, targets them for secretion. The signal peptide is cleaved off in the endoplasmic reticulum. Pre-proproteins have both sequences, inhibitory and signal, still present. In protein synthesis, a succession of transfer RNA molecules charged with appropriate amino acids are brought together with a messenger RNA molecule and matched up by base pairing through the anticodons of the transfer RNA with successive codons of the messenger RNA. The amino acids are then linked together to the extend the growing protein chain, and the transfer RNAs no longer carrying amino acids are released. This whole complex of process is carried out by the ribosomes, formed of two main chains of RNA called ribosomal RNA, rRNA, and more than 50 different proteins. The ribosome latches onto the end of a messenger RNA molecule and moves along it, capturing loaded transfer RNA molecules and joining together their amino acids to form a new protein chain. Protein biosynthesis, although very similar, is different for prokaryotes and eukaryotes. We now talk in general about the three major levels of protein biosynthesis: transcription, translation and post-translation, that is, the events following protein translation. In transcription, messenger RNA chain is generated with one strand of the DNA double helix in the genome sequence serving as a template. This strand is called the template strand. Transcription can be divided into three stages: initiation, elongation and termination. Each is regulated by a large number of proteins such as transcription factors and coactivators that ensure that the correct gene is transcribed. Transcription occurs in the cell nucleus where the DNA is held. The DNA structure of the cell is made up of two helix made up of sugar and phosphate held together by hydrogen bonds between the bases of opposite strands. The sugar and the phosphate in each strand are joined together by stronger phosphodiester covalent linkage bonds. The DNA is unzipped, that is, disruption of hydrogen bonds between different single strands, by the enzyme helicase, leaving the single nucleotide chain open to be copied. RNA polymerase reads the DNA strand from the 3' site to the 5 prime end, while it synthesizes a single strand of messenger RNA in the 5 prime to 3 prime direction. The general RNA structure is very similar to the DNA structure, but in RNA, the nucleotide uracil takes the place that thymine occupies in DNA. The single strand of messenger RNA leaves the nucleus through the nuclear pores and migrates into the cytoplasm. The first product of transcription differs in prokaryotic cells from that of eukaryotic cells, as in prokaryotic cells the product is messenger RNA, which needs no post-transcriptional modification, whereas in eukaryotic cells the first product is called primary transcript that needs post-transcriptional modification, that is capping with 7-methylguanosine tailing with a poly A tail to give hnRNA, that is heterophil nuclear RNA or heterogeneous nuclear RNA. hnRNA then undergoes splicing of introns, that is non-coding parts of the gene, via spliceosomes to produce the final messenger RNA. Translation. The synthesis of proteins from RNA is known as translation. Translation occurs in the cytoplasm where the ribosomes are located. Ribosomes are made up of small and large subunit that surrounds the messenger RNA. In translation, messenger RNA is decoded to produce a specific polypeptide according to the rules specified by the trinucleotide genetic code. This uses a messenger RNA sequence serving as a template to guide the synthesis of a chain of amino acids that form a protein. Translation proceeds in 4 phases: activation, initiation, elongation and termination, all describing the growth of amino acid chain or polypeptide that is the product of translation. In activation, the correct amino acid is joined to the correct transfer RNA. While this is not in the technical sense a step in translation, it is required for translation to proceed. The amino acid is joined by its carboxyl group to the 3' OH of the transfer RNA by an ester bond. When the transfer RNA has an amino acid linked to it, it is termed charged. Initiation involves the small subunit of the ribosome binding to 5' end of messenger RNA with the help of initiation factors IF, other proteins that assist process. Elongation occurs when next amino acid tRNA, charged tRNA in the line, binds to the ribosome along with GTP, that is guanosine triphosphate, and an elongation factor. Termination of the polypeptide happens when the A site of the ribosome faces or stop or nonsense codon UAA, UAG or UGA. When this happens, no tRNA can recognize it, but releasing factors can recognize nonsense codons and cause the release of the polypeptide chain. The mature messenger RNA finds its way to a ribosome where it gets translated. In prokaryotic cells which have no nuclear compartment, the process of transcription and translation may be linked together without clear separation. In eukaryotic cells, the site of transcription, that is the cell nucleus, is usually separated from the site of translation, that is the cytoplasm. So, messenger RNA must be transported out of the nucleus into the cytoplasm where it can be bound by ribosomes. The ribosome reads the messenger RNA triplet codons, usually beginning with an AUG, adenine, uracil, guanine, or initiator methionine codon downstream of the ribosome binding site. The complexes of initiation factors and elongation factors bring amino acylated transfer RNAs into the ribosome messenger RNA complex, matching the codon in the messenger RNA to the anticodon on the transfer RNA. Each transfer RNA bears the appropriate amino acid residues to add to the polypeptide chain being synthesized. As the amino acids get linked into the growing peptide chain, the chain begins folding into correct conformation. Translation ends with a stop codon which may be a UAA, UAG or UGA triplet. The messenger RNA does not contain all the informations for specifying the nature of the mature protein. The nascent polypeptide chain released from the ribosome commonly requires additional processing before the final product emerges. For one thing, the correct folding process is complex and vitally important. For most proteins, it requires other chaperone proteins to control the form of the product. Some proteins then excise internal segments from their own peptide chains, splicing the free ends that border the gap. In such process, the inside discarded sections are called inteins. Other proteins must be split into multiple sections without splicing. Some polypeptide chains need to be cross-linked and others must be attached to the cofactors such as heme before they become functional. The capacity of disabling or inhibiting translation in protein biosynthesis is used by some antibiotics as N-isomycin, cycloheximide, chloramphenicol, tetracycline, streptomycin, erythromycin, puromycin, etc. Now we talk about post-translation, that is the events following protein translation. The events following biosynthesis include post-translational modification and protein folding. During and after synthesis, polypeptide chains often fold to assume the so-called native, secondary and tertiary structures. This is known as protein folding. Thus, after protein amino acid sequences have been translated from the nucleic acid chains, they can be edited by appropriate enzymes. Although this is a form of protein affecting protein sequence not explicitly covered by the central dogma, there are not many clear examples where the associated concepts of the two fields have much to do with each other. Now let us see the difference between eukaryotic protein synthesis versus prokaryotic protein synthesis. Eukaryotic messenger RNA molecules are monocistronic, that is containing the coding sequence only for one polypeptide. In prokaryotes, messenger RNA molecules are polycistronic, containing the coding sequence of several genes of a particular metabolic pathway. In eukaryotes, protein synthesis occurs in the cytoplasm, whereas in prokaryotes, protein synthesis begins even before the transcription of messenger RNA molecule is completed. This is called coupled transcription translation. In eukaryotes, most of the genes have introns or non-coding sequences along with exons or coding sequences. The exons are joined together and introns are removed during the messenger RNA processing, whereas prokaryotes do not have introns except archaebacteria. Therefore, messenger RNA processing is not required. The primary messenger RNA transcript in eukaryotes undergoes processing and splicing to change into a functional messenger RNA. In prokaryotes, splicing of messenger RNA transcript does not occur. In eukaryotes, messenger RNA molecules are modified by the addition of a 5' G cap formed of methylated guanosine triphosphate. No such cap is formed at the 5' end of bacterial messenger RNA. A poly A tail formed of about 200 adenine nucleotides is added at the 3' end of messenger RNA in eukaryotes, whereas no poly A tail is added to bacterial messenger RNA. In eukaryotes, 5' capping initiates translation by binding the messenger RNA to small ribosomal subunit, usually at the first codon AUG. In bacteria, translation begins at an A codon preceded by a spatial nucleotide sequence. In eukaryotes, the first amino acid methionine entering the ribosome is not formulated. In prokaryotes, the first amino acid methionine is formulated into N-formyl methionine. In eukaryotes, the number of initiating factors is much more than prokaryotes. About 10 initiating factors IFS have been identified in reticulocytes and RBC. These are elongation factor I, elongation factor F2, F3, F4, F5, F6, F4B, F4C, F4D and F4F. Whereas 3 initiating factors found in prokaryotes are prokaryotic initiating factor PIF1, 2 and 3. In eukaryotes, small subunit of ribosome, that is the 40S, gets dissociated with the initiator amino acyl transfer RNA, that is MIT RNA MET, without the help of messenger RNA. The complex joins the messenger RNA later on. In prokaryotes, 30S subunit first complexes with the messenger RNA, that is the 30S messenger RNA complex, which then joins with formyl methionine tRNA. Here we need to talk about other facts associated with protein biosynthesis. Reverse transcription. Reverse transcription is the transfer of information from RNA to DNA, the reverse of normal transcription. This is known to occur in the case of retroviruses such as HIV, as well as in eukaryotes in the case of retrotransposons and telomere synthesis. It is the process by which the genetic information from RNA gets transcribed into new DNA. RNA replication. It is the copying of one RNA to another. Many viruses replicate this way. The enzymes that copy RNA to new RNA, called RNA-dependent RNA polymerases, are also found in many eukaryotes where they are involved in RNA silencing. RNA editing. Here an RNA sequence is altered by a complex of proteins and a guide RNA, that is gRNA, which could be seen as a RNA to RNA transfer. Direct translation from DNA to protein. Direct translation from DNA to protein has been demonstrated in a cell-free system, that is in a test tube, using the extracts from E. coli that contained ribosomes but not intact cells. These cell fragments could synthesize proteins from single-stranded DNA templates isolated from other organisms, example mouse or toad, and neomycin was found to enhance this effect. However, it was unclear whether this mechanism of translation corresponded specifically to the genetic code. Now we make a special mention of the central dogma of molecular biology. The central dogma of molecular biology is an explanation of the flow of genetic information within a biological system. It was first stated by Francis Crick in 1956 and restated later on. The central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information. It states that such information cannot be transferred back from protein to either protein or nucleic acids. The central dogma has also been described as DNA makes RNA and RNA makes protein, a positive statement which was originally termed the sequence hypothesis by Crick. However, this simplification does not make it clear that the central dogma as stated by Crick does not preclude the reverse flow of information from RNA to DNA, only ruling out the flow from protein to RNA or DNA. Crick's use of the word dogma was idiosyncratic and has been controversial. The dogma is a framework for understanding the transfer of sequence information between sequential information carrying biopolymers, and the most common or general case in living organisms. There are three major classes of such biopolymers: DNA and RNA, both nucleic acids and proteins. There are 3 by 3, that is 9 conceivable direct transfers of information that can occur between these. The dogma classes these into 3 groups, that is 3:3. General transfers believed to occur normally in most cells. 3 spatial transfers known to occur but only under specific conditions in case of some viruses or in a laboratory. And 3 unknown transfers believed never to occur. The general transfers describe the normal flow of biological information. DNA can be copied to DNA, that is DNA replication. DNA information can be copied into messenger RNA, that is transcription, and proteins can be synthesized using the information in the messenger RNA as a template, that is translation. The biopolymers that comprise DNA, RNA and polypeptides in linear polymers, that is each monomer is connected to at most 2 other monomers. The sequence of their monomers effectively encodes the information. The transfer of information described by the central dogma ideally are faithful, deterministic transfers, wherein one biopolymer's sequence is used as a template for the construction of another biopolymer with a sequence that is entirely dependent on the original biopolymer's sequence. Now we talk about the general transfers of biological sequential information. Here the table of 3 classes of information transfer suggested by the dogma shows: general case, DNA to DNA, DNA to RNA and DNA to protein. The special case, RNA to DNA, RNA to RNA and RNA to protein, and in unknown protein to DNA, protein to RNA and protein to protein. We also now become familiar with some terms as inteins. An intein is a parasitic segment of a protein that is able to excise itself from the chain of amino acids as they emerge from the ribosome and rejoin the remaining portions with a peptide bond in such a manner that the main protein backbone does not fall off. This is a case of a protein changing its own primary sequence from the sequence originally encoded by the DNA of the gene. Additionally, more inteins containing a homing endonuclease or HEG domain, which is capable of finding a copy of the parent gene that does not include the intein nucleotide sequence, on contact with the intein free copy, the HEG domain initiates the DNA double-stranded break repair mechanisms. This process causes the intein sequence to be copied from the original source gene to the intein free gene. This is an example of protein directly editing DNA sequence as well as increasing the sequence's heritable propagation. Epigenetics and methylation. Variation in methylation states of DNA can alter gene expression levels significantly. Methylation variations usually occur through the action of DNA methylases. When the change is heritable, it is considered epigenetic. When the change in information status is not heritable, it would be a somatic epitype. The effective information content has been changed by means of the action of a protein or proteins on DNA, but primary DNA sequence is not altered. Prions. Prions are proteins of particular amino acid sequences in particular conformation. They propagate themselves in the host cells by making conformational changes in other molecules of protein with the same amino acid sequence, but with a different conformation that is functionally important to the cell. Once the cell protein has been reconformed to the prion configuration, it no longer functions in the way that the cell requires, and its turn it can infect cells and reconfigure more functional molecules of that sequence into harmful form, example mad cow disease. In some types of prion in fungi, this change is continuous and direct. The information flow is protein to protein. This does not represent a transfer of information, but the prion interactions leave the sequence of the protein unchanged. Lastly, we discuss the emerging ideas. It is becoming increasingly clear that in reality, the concept of the central dogma of molecular biology is not entirely accurate insofar as it puts emphasis on proteins as the mediator of biological function. We know that 80% of human genome is transcribed even though only 1% codes for the proteins. While it is possible that this may be simple transcriptional noise, it seems to be an unlikely waste of cellular energy resources, and considering the major role played by RNA in regulation of gene expression, it may well have a role. Current research focuses on investigating the function of non-coding RNA, that is RNA that does not follow the dogma trend and does not code for polypeptides. Moreover, the precise meaning of information in this framework is often overlooked.
Now let us summarize the topic protein biosynthesis. Protein biosynthesis is a biochemical process in which proteins are synthesized from simple amino acids in several steps with the help of information stored in the DNA. First of all, activation of all amino acids and formation of amino acyl tRNA occurs here. 20 different proteinogenic amino acids are activated as they all are esterified with a tRNA specific to them. This takes place in cytoplasm by amino acyl tRNA synthetases, a group of enzymes which are specific only to a certain amino acid respectively. The activation energy is supplied by ATP consumption. Secondly, initiation of the chain formation and binding to ribosome subunit occurs. by binding the mRNA and the first amino acyl tRNA to a free 30S ribosome subunit, an initiation complex is formed to which the 50S subunit is attached in the second step. The first amino acyl tRNA begins the process as an N-acyl derivative to make sure that the synthesis of polypeptide chain always starts at the beginning of a genetic message. Then the chain elongation occurs, whereby the peptide chain is elongated by the continuous attachment of amino acyl residues. These residues are translated by amino acyl tRNA esters, each determined by a codon in the mRNA. After each new peptide is attached, both mRNA and the peptidyl tRNA chain are moved to a little along the ribosome in order to properly position the next codon. The necessary reactive power is supplied by GTP consumption. Lastly, the chain termination and the release from the ribosome occurs here. The polypeptide chain is terminated by 3 special stop codons. Eventually, the completed chain separates from the ribosome. The release of the polypeptide tRNA from the ribosome is initiated when the stop codon is reached by a specific protein factor which is attached to the ribosome and hydrolytically cleaves the ester bond between the polypeptide and tRNA. Thereupon, the 70S ribosome leaves the mRNA in the free form. It can enter a new cycle if it first dissociates in its 50S and 30S subunits, for which one specific initiation factor is required. Thank you.